Buenos días, good morning everyone. I have to thank to the organizers to give me the possibility to present some of our more recent work. So we are starting directly with the presentation. So probably in this room I don't need to do a full presentation of what is Alzheimer, so I'm sorry for that, but I had to remember some of the data that I am going to need for the presentation. So just to remember some of our most important data from Alzheimer, I have to just to say that Alzheimer present two hallmarks that are absolutely diagnostic. So okay, we can follow because I think that I don't have the presentation is not in my computer. It's gone. No, doesn't matter really. So Alzheimer is characterized by two different histopathological markers. One is the accumulation of the beta amyloid peptide in the senile plaques, and the second one, which is the intercellular accumulation of the neurofibrillary tangles. That it's basically made by tau, phosphorylated tau. Just to remember, as I told you, some of the data, the amyloid peptide is generated by proteolytic cleavage from the amyloid precursor protein. It's a housekeeping protein, and the process of proteolysis is conserved in all cell types. And from one of the two different pathways, we can obtain the peptide. The pathway is called the amyloidogenic pathway, and the consecutive proteolytic activity of the beta secretaries and the gamma secretaries generates some peptide with 40, 42, and 43 amino acids. So the second markers in Alzheimer is the tangle that are made from the tau protein, almost the full-length protein. In that case, the protein is almost exclusively neuronal protein, and is developmental regulated by phosphorylation, among other different things. And the phosphorylation appears to be very, very high in development and in Alzheimer's disease too. However, the accumulation and degradation of protein is not a specific characteristic of Alzheimer, and in other pathologies, in other neuropathologies, appears not only tangle, but the accumulation of different proteins that can be accumulated in the extracellular matrix or in the intercellular neurons, like many of you know. Alpha-synuclein, huntingtin, and the prion proteins that I have to mention. And I would like to give you some of the data of the prion protein because I need it for the presentation. So just remember, the prion disease is a very, very well-known disease, and I mean, was considered completely different because in some moment was the only that we can say that this is an infection of a protein. The accumulation of the protein is the PRP protein, that is the cellular component of this element, and the pathology appears not only in humans like a Creutzfeldt-Jakob, but even in sheep, in cows, cats, and many other mammals. The prion protein is just a GPI membrane-anchored protein that is mostly neuronal with no clear function so far, and the conversion to the pathological one apparently is a conversion with a change in the second structure to increase in the beta sheet component, and most important, with a resistance of protease, which is a characteristic of the accumulation of this protein. Apparently, we thought a long time ago that this is the only protein that is able to recruit, to infect by recruiting the normal protein, And in this scheme, you can see just the hypothetical recruitment. So the pathological one that is called PRPSC from Scrapie is able to recruit the normal protein that's in the membrane, that is in the membrane, the PRPC, and for mechanisms that is not completely clarified, probably with helping of one different membrane cofactor of, or maybe in one cellular compartment not completely clarified. The normal one is converted to the pathological one, and this is a chain of conversion which we thought that was exclusive in this pathology. On the other hand, the PRP cellular one is, as I told you, one cellular protein, neuronal protein that interacts with different membrane proteins. In particular, I want to remember that the PRP is able to interact with APP in the membrane. So we had different papers demonstrating, suggesting that the interaction is important for the synaptic transmission for the dysfunction of synaptic transmission of amyloid, but this issue is still an open question. So just like a general consideration, many of the aggregation of the protein presented similar rules. So the misfolded protein is accumulated in some neuronal structure and are able to aggregate initial generating oligomers and then polymers, and then the accumulation, depending on the type of protein, can be in the nucleus, in the cytoplasm, or in the extracellular matrix of the brain. So for that reason, many of this aggregation of protein are considered prion-like structure, not only but the kinetic of polymerization, but even for report like that, in what Claudio Soto lab showed clearly that the injection, the ICV injection of amyloid samples is able to generate the amyloidosis inside a transgenic mice. So the demonstration strongly suggests that the amyloid behave like a prion-like disease. It's not only demonstrated for amyloid. So in that point, we're trying to analyze two different things, and that was an initial collaboration with a group of CIBERNED, that is Tony Del Rio groups, and another group outside of CIBERNED. And the question that we're trying to answer was, there is a possible interaction between two different amyloids. If we consider that amyloid have similarity with a polymerization process, the question is, the presence of prion can be modified, or the infection of prions can be modified by different levels of amyloid deposits in the brain of a mice. And the second question was the opposite one, which is may the amyloid accumulation be modified or be sensitive to the presence of PRPC in the neuronal brain? For approach these two different questions, we organized different transgenic mouse lines containing different amount of PRPC, the cellular component of the neuron, and we use the human version of mutant APP and PS1 to make the proper combination with both mice. The parental lines are those. This is the APP and PS1 mutant. This is the little mate. This is the wild type with the same background that the APP mice. This one is the transgenic mice, the knockout mice with the deletion of PRP. So they don't contain PRP, obviously they don't contain the APP. And that one is the transgenic mice expressing different amounts of PRP-C. The expression is around 4-6 fold levels from the normal situation. So the breeding protocol was that, and we obtained that different mice combination from in one case we using two different back crossing, and in another only one. And just this is the mice that we generated. Basically, you have no PRP. This is the knockout for PRP with or without the APP. One copy of the PRPC with, again, one or two with or without APP. Then the wild type, that is two copies of the PRP and no APP. And the transgenic lines showing, again, 3, 4, 6 copies of PRP and with or without the APP transgenic. The first thing that we checked, because it was published that the mouse lines containing APP have a low level of viability. The first one that was reported a couple of years ago, we showed that in our cell lines, in our mouse lines, the viability was not really modified, and in all cases, the presence of APPPS1 reduced just a little bit the viability, but it's not like reported that is completely different. The second point was we're trying to answer the question: the presence of APP modified the infectivity of the prions. And the experiment was done with using two different prions. One, which is the strain 22L, which is a prion mouse adapted from many years and used in many laboratories. And the second one was a bobbing prion modified and adapted to the mouse brain. And in both cases, you can see that each strain have a specific survival time, but the presence of amyloid in the brain does not modify statistical significant the survival. Again, with the bobbing strain happens similar and it's not statistical significant. And the second type of experiment was just to show with the using 22L that the prion is only sensitive to the amount of PRPC in the presence of the brain. So mouse without PRPC never is infected. Then with one copy or two copies or a lot of 6-4 copies, the survival time is shorter, but they respond only to the presence of PRP and not to the presence of the amount of amyloid in the brain. So that is the control to show that each strain present, injected, and recovered from the brain of infected mouse are not modified by the context of a brain, independent of the genetic background that we are using. So we move to the second question, is the contrary. So is the amyloid deposit modified by the PRP, by the PRPC, by the cellular component of the prion protein? Again, the protocol that we used was the similar one. We generated the mouse lines, and then we wait until 3, 6, and 9 months, and the general rule of processing the brain was something like that. Half of the brain are going to be used for immunohistochemistry. The second one is used for ELISA and the biochemical studies. The data, the most important data was that we cannot detect any difference from 3 to 6 months independent of the amount of PRP. However, after 9 months older, the mice increase the amount of amyloid 140 and 142 in a nice correlation with zero copy copies of PRP, 1, 2, and this is the 4, 6 element. So the amount of 140 determined by ELISA are clearly increasing in those old animals. So just to compare, to check and to confirm this data, we use different antibodies. We check that the APP, the transgenic, is not sensitive modified along aging. The control obviously don't have the expression of the transgene. That this animal are the transgenic for PRP, 2 copies of them, 0 copies. So, and in all cases, when the expression of PRPC is high, the monomer and the oligomer are increasing after 9 months of aging. That was confirmed by immunohistochemistry, so we compare and determine the level of amyloid accumulation, let's say senile plaques present in the brain, and the quantification shows, again, a nice correlation between 0, the wild type, and the transgenic. Clearly, the amyloid deposits and the senile plaques are increasing in a clear correlation with the amount of amyloid that we are determining in the brain. The second analysis that we try to do is to search some information, what is the meaning of this extra accumulation of the amyloid in the brain, and we check, obviously, many of the pathways that we consider can be modified by the expression of by amyloid or by monomers. So we check for different phosphorylation of taus and different kinases that can be implicated or has been reported to be implicated in Alzheimer. So, and basically, we don't detect any major difference, or we don't detect difference statistical significant among these different mice. In some cases, because the intra-mouse difference is too high, probably because there are kinases implicated in normal metabolism. So the difference from one mice to another, in some cases, there are broader from the same offspring. So the difference between mice don't permit to have a conclusion of what is happening with these different kinases. And the same for different, we analyze inactive protein like PSD 95, synapsin. In some cases, that this protein has been reported to be modified in this specific mouse model. As you can see, no major difference were detected at this time. The only difference that we check, we analyze next the phosphorylation of tau. We train almost all the antibodies that we have, pH1, tau1, 80, 100, and only we detect difference with the antibody against the tyrosine 18. As you can see here, the transgenic PRP TG20 have a clear increase of the tyrosine phosphorylation of tau. And finally, that is the last data that we have. Has been reported that Alzheimer may have the autophagic pathway modified. We don't know if this modification is a direct modification of the pathology, or if this is a response of the neuron against the pathology. So that is something that has to be confirmed and work. And what we did was the analysis of one of the major systems that controlling autophagy, which is the mTORC1 kinase. This kinase is able to control B17S6 kinase and the control of transduction protein, a translation protein, and by a complex system with the PI3 kinase of class 3 is able to control some elements that are controlling the phagophore and is a system that controls ATG12.5, ULK, the conversion of LC3 to LC32, which is a marker of the autophagy. We check almost all the components, and we detect only an increase in LC32, which is quantified here, showing probably that the accumulation of amyloid triggered by the increasing presence of PRP increase the level of autophagy in these neurons, or at least in these brains. So, just like conclusions, the infectivity of prions is not different depending of amyloid. So, the prions infection only recognize the amount of PRP protein in the brain. Doesn't matter if you have a different amount of amyloid in the brain. So, however, in the other way, the amyloid burden is modified. It's affected by the PRPC, the present. When we have a lot of amount of PRPC, the amyloid, the accumulation of amyloid is clearly and is evident. The increasing of amyloid in TG20 is age-dependent, which maybe suggests that we have another different, we have an extra element which is age-dependent, something that has to be with the aging factor that we are trying to identify or to analyze. And finally, the presence of APP, PS1 and TG20 apparently increase not only the amyloid in the brain, but trigger a response in the neurons that in part is clearly an increase of autophagy, which probably suggests that it's trying to get rid of the amount of amyloid that we are overexpressing. No more. Just the most important slide. The people that did the work was the prion business was performed in the Juan Maria Torres lab in the CISA, because it's a P3, high security level P3 animal room facility. And the remain of the data was made in the Jose Antonio del Rio in Barcelona, in my lab in Madrid, and the responsible were Lara Ordóñez and Rosalina Gavín. Thank you very much for attention.
And you should repeat the question.
Okay, the question is that the question was is the accumulation or the production of the amyloid, which is different, and I think that the data suggests that there is the production of the amyloid which is increasing, and the accumulation too, because we are detecting more senile plaques. And the second question was the distribution. We check in the cortex and in the hippocampus, and the distribution is almost similar. Probably, I think, if I remember the data, it's more evident in the hippocampus, but it's similar. It's increasing too in the cortex, which I don't know. I mean, I don't know if the differential distribution is important or not, because this is a transgenic control by PRP promoter, so the distribution is under control, the promoter, So the significance of the accumulation, of the local accumulation, maybe is something to discuss.
I thought the autophagy observation was very interesting, and I just wondered if you have looked at all at the subcellular differences in autophagy. Are you sure that the LC3 production is in neurons? There's some thought that the activation of the mTOR pathway may be different in different cell types in the brain.
We didn't check in the brain, so we are moving to the neuronal culture. So we now we are starting with a specific project analyzing the autophagy in neurons, and we know that the accumulation of LC3 in neurons, in transgenic neurons, in cell culture is different from the wild type. So